Good morning, friends. Welcome to this short lecture session. I am Dr. Ayaz Malik, Associate Professor, Department of Biochemistry, and I am going to talk on organization of DNA. Well, the objectives of this lecture would be to describe histones, list the steps of organization of DNA, and explain the organization of DNA into a chromosome. Well, starting with the introduction, well, we all know what DNA is. DNA is a double helical structure molecule which is made up of 4 base pairs, and this DNA has a diameter of about 2 nanometers, and it is actually the carrier of the genetic information. This DNA is organized into a highly specialized structure called as the chromosomes, and it is present in the nucleus of the cell. Now, if we try to stretch the DNA, if we pull the entire DNA and stretch it, it would measure about 2 meters, whereas the diameter of the nucleus is around 6 micrometers. Now we can imagine trying to put a string which is about 2 meters long into a box which is about 6 micrometers small. This is where the entire concept of organization of DNA comes into play. So before we move on to the organization of DNA, we should understand are these chromosomes. These chromosomes are actually made up of threads of chromatin, and these threads of chromatin are composed of mainly the DNA, because that is what is getting organized, along with proteins which will help it to get organized, and there's a small amount of RNA as well. Now these proteins are mainly of two types. One called as the histone proteins, which are involved in the organization of DNA. These are basic proteins. They have a positive charge on them, and all the other proteins are classified as the non-histone proteins, which actually play a role in the regulation of gene. They are involved in the translation transcription process. So before we move on to the organization, we should understand what are these histones. As already mentioned, these are small proteins having a molecular weight of about 11 to 20 kilodaltons. They are rich in basic amino acids, especially arginine and lysine. As a result of this, they have a positive charge on them. And these histones actually react in a very specific manner. They undergo modification in the form of acetylation, phosphorylation, ribosylation, and their interaction with the DNA varies at various stages. Coming on to the types of histones, histones is not a single protein but a group of proteins, and we have the H1 protein, H2A, H2B, H3, and H4. Now it is all these different types of histone, they interact together and they bring about their action. Coming on to the stages of the organization of eukaryotic DNA into a chromosome, there are 4 stages. The first stage is the formation of a nucleosome, followed by formation of a 10 nanometer fibril. Then there is formation of 30 nanometer fiber. Then there will be formation of loops and domain, and finally there will be formation of the chromosomes. Now before we move on to the steps of organization of DNA, we should understand how these histones interact with each other. As already mentioned, there are different types of histone proteins. 2 molecules of H2A, 2 molecules of H2B, and a tetramer which has 2 molecules of H3 and H4. They interact with each other and form what is called as the histone core or the octamer. Now the formation of this octamer is very important because this is the molecule that is going to start the entire organization of DNA. Well, now we move on to the steps of organization of DNA. The DNA, which is around 2 meters long, it starts winding around the histone core. As we can see here, this is the DNA which is winding around the histone core in such a way that it takes around 2 turns around each octamer of the histone core molecule and continues. This interaction between the DNA and the histone core gives rise to the nucleosomes, which is the basis of the organization of DNA. The same nucleosome is spread throughout the DNA, and it gives it appearance called as the bead and string appearance. Now as you can see here, this is one nucleosome, the second nucleosome, third nucleosome, and so on. Between these nucleosomes there is a stretch of DNA which is free, and that is called as the linker DNA. And this nucleosome is stabilized by a protein, that is the histone H1. So this molecule, where we have a DNA wound around the histone called as the nucleosomes, the linker DNA, this gives rise to the second step, that is formation of a 10 nanometer fibril. Continuing further, this 10 nanometer fibril undergoes further winding, further coiling in such a way that the nucleosomes are wound around each other. Now each turn will have around 6 nucleosomes wound around each other, and the diameter of this structure now is 30 nanometers, so it is called as the 30 nanometer fiber. Once this 30 nanometer fiber is formed, the process of coiling and winding continues till the entire DNA is thrown into a looped structure. Now this loop structure needs to be stabilized, and that is done by a protein called as the scaffold protein. This scaffold protein actually anchors the entire loop structure to it and prevents it from straightening. Now the diameter of this structure right now is 300 nanometers. Once the loop structure is formed, there will be further condensation and coiling, and the DNA starts excessive coiling over each other and forms a loops and domains. And now the diameter has increased to about 700 nanometers. So now once these loops are formed, there will be further compaction, and the coiling continues and continues till this entire stretch of DNA, which was 2 meters long, gets condensed into a chromosome having a diameter of about 1400 nanometers. Now let's try to summarize how this DNA is condensed into a chromosome. I told you there are 4 steps, 4 stages. In the first stage, what happens is that there will be formation of nucleosomes, where a DNA molecule will interact with the histone core and forms the nucleosomes. These nucleosomes are stabilized by the H1 protein, and they are separated by a stretch of DNA called as the linker DNA. This step is called as the formation of 10 nanometer fibril. There will be further coiling, winding, and now the nucleosome starts coiling over each other and forms a 30 nanometer fiber. The coiling condensation continues, and the DNA is thrown into loops which are stabilized by the scaffold protein. There will be further coiling, and finally the DNA will be condensed into a chromosome which will be packed into the nucleus of a cell. With this, I would like to thank you all for attending my lecture. Thank you very much and have a nice day.